Okay, plasma membrane, let's get into it. Basically, plasma membrane separates the internal environment of the cell to the external environment of the cell. It's this little barrier here that separates it. It's very important because it regulates all of the interactions between the inside of the cell and the outside. So this is exchange of nutrients, this is getting water, this is the concentration of oxygen. It's very important. The structure of the plasma membrane is demonstrated by the fluid mosaic model, which was created by some boring people. Basically, the fluid bit refers to the phospholipid bilayer, and the mosaic model refers to all the stuff in the middle. So I'll talk a bit more about this, but basically the fluid bit is these molecules here that create a phospholipid bilayer, which I'll talk about in just a second. Don't stress. And the mosaic bit is kind of all the stuff that's embedded into it. It's the sugars and the other molecules and that and that and that. It makes a mosaic. That's why it's called that. The phospholipid bilayer, that's this bit, the fluid section. It is the main boundary bit of the plasma membrane. So all of this all together is the plasma membrane. But just this section here and this section is the phospholipid bilayer, or the fluid component. The phospholipid bilayer meaning two layers, where we've got this and this. The phospholipid bilayer is made up of phospholipid molecules. What the heck does that mean? Excellent question. This is a phospholipid molecule. This is a phosphate head, and these are fatty acid tails. So these fatty acid tails, they hate water, right? They are hydrophobic. If you think of hydro as water, hydrophobic as like you're scared of, you hate water. And they are non-polar, which you guys won't know what that means because you wouldn't learn about it in chemistry and it was never explained to us. But basically this means that they are neutral and they only attract other neutral molecules, right? Water is polar. So it kind of makes sense that it doesn't like water because water has a charge to it, which means that these non-polar tails don't like the water. On the other hand, the phosphate head is hydrophilic. Philic meaning love, right? It loves water. And it is polar, which means that the charge is uneven. It's like slightly positive or slightly negative, whereas here it's neutral. This means that because of its polarity, it likes water and it attracts water. So when you line them all up in a boundary, right, you have this area here that is made up of these tails that doesn't like water, so it does not let water in freely. However, here it does like water, so it attracts water. This works out really well for the cell because basically if this is a cell, oh, that's a really good circle. That's also a good circle. Loki good at drawing circles. Anyways, If this is a cell, it means that if all of this is water, lots of water around here, the outside will attract the water because the cell needs water. However, the inside of the cell will only let water in if it needs it, which keeps the cell regulated, and this is really, really important. So, this middle section made of the tails don't allow anything to freely pass through if it's polar. This is things like potassium or sodium, right? Because they all have this positive polarity, right? Or if it's negative, they all have a negative thing. In eukaryotes, us, the phospholipid bilayer also contains cholesterol, which a lot of the time we think, oh my gosh, bad, your cholesterol. No, we actually do need this because it stabilizes the bilayer at different temperatures, which is really important because it means that we are less vulnerable to external changes, right? So at high temperatures, it stabilizes the plasma membrane, and at low temperatures, it packs the membrane together, making it firm. So here, when the temperature increases, what it does is it actually makes it harder for the plasma membrane to melt or boil, right? Imagine how terrible it would be if for some reason on a 40-degree day we just like melted, right? Basically, it increases the melting point, which means that the cells need to reach a higher temperature in order to melt, and at lower temperatures it makes the cell more firm. Another really important part of the cell and us in general is proteins. So they are a vital aspect of the plasma membrane, but they are embedded into the membrane. So the proteins make up the mosaic component of the plasma membrane. Now, different proteins have different functions, and I'm not sure if you guys remember, but it was a very helpful acronym to help us, or help me, or whatever, help me remember what the 6 key functions of a protein was, and that is TRACY. So, TRACY. T. Proteins are really important for transporting materials or minerals in and out of the cell. R. They are important for cell-to-cell recognition. So this is if, for example, in the immune system, how does a cell tell the difference between good bacteria that is from us and bad bacteria that is not from us and that needs to be killed? The proteins. I think we'll talk a bit more potentially in depth about how this actually functions, but that's really important. Proteins help the cells recognize each other. A, this is attachment to the cytoskeleton. So this is what keeps the cell structurally sound. We want this. C, this is cell signaling. So this is like hormone communication, the cells talking to each other. Again, very, very important. I is intercellular joining. This is really important. This is like the cells, if these are the plasma membranes, right? These are the cells like actually connecting to each other, which is, you know, how I'm together. So that's very important. And finally, E. This is enzyme activity. So this is things like breaking excess products down or nutrients or whatever. So Tracy, this is a super, super helpful acronym for the functions of proteins, and I hope you guys can see that they are very important. So, there are two different types of protein in the phospholipid bilayer. So, if we draw the phospholipid bilayer, or at least one side, right, we have the tails. I'm sure, hopefully you understand what I mean, right? There are two and a half kinds of proteins in the phospholipid bilayer. So I'll just do this. So we've got integral and peripheral. Integral or integral, actually, I don't know if I'm saying that right. Then once you have integral, you have also got transmembrane. So you've got another either integral or transmembrane, right? Integral proteins, these are proteins that are permanently part of the phospholipid bilayer, whereas peripheral is only temporarily part of the phospholipid bilayer. All integral proteins are permanent. However, transmembrane proteins span across the entire space. So I'm going to start to draw them. We'll go with, because I'm trying to show you that this is also part of the mosaic, but transmembrane proteins go this whole way, right? If this is the other side of the phospholipid bilayer, right? It goes across the whole way. Integral, just straight integral, they are still part of the phospholipid bilayer permanently. However, they don't span the whole way across. Do you see that? Now, the difference is that with peripheral proteins, they actually don't even cross this space. So, see, remember how this space I was talking about is hydrophobic? If you think of it, peripheral proteins, they're not that special, they don't get to go through the whole thing, so they actually just either sit outside of the plasma membrane or they attach to another protein to help their function. However, because they are only temporary, they don't really get to be part of it, they don't get to be in, so they only attach to the actual phosphate heads or to another protein. So I hope this makes sense. That's kind of all to it. We're going to look at carbohydrates or sugars. So this is like sugar, glucose, whatever. These guys are really big and they are also very important. They attach themselves to either proteins or lipids to kind of get into or on or through the plasma membrane. So there are two kinds. So remember glucose, we have glycoproteins, which is pretty self-explanatory. We've got the carb and we've got the protein. And we also have glyco, so carb, and lipid, which is a fatty molecule. Now, a lipid, is actually made up of a phosphate head and two little fatty tails. This is exactly what the plasma membrane is made out of. So this glycolipid, the glyco kind of always looks like this, right? We've got that. It's that plus the lipid. Whereas here, the protein could look, I don't know, just like a little thing like this. The glyco bit is still the same, but altogether, the molecule is different. Okay, so a really good example of how these can be used is with the white blood cells. So these are the guys that fight off bacteria. They recognize whether or not something is part of your immune system or not. Now, what happens is they have, say this is a white blood cell, right? They have specific little receptors on them. And if you are good to go, you'll have a specific glycoprotein that matches perfectly with this, and the cell will go, yep, cool, you're all good to go. I like to think of this as a PTO officer, right? And it's checking whether or not you have your right Mikey, okay? However, if you have a white blood cell and this is a bad cell, this is bacteria or whatever, it actually won't have this right glycoprotein, and the white blood cell or the PTO officer will say, nah, got to give you a fine, or it'll start to kill it, right? So that is where the glycoprotein is really helpful. So I just think that's helpful.